and thank you for joining us in the Salon. I'm Adam Smith. I'm the Program Director for Dementia Researcher, and I'm delighted to be hosting today's meeting, which is all about clinical trial design. As you probably know, in these weekly sessions, we explore research, career topics to get some new perspectives, to educate and entertain, and hopefully give you some new ideas and get you a chance to hear yours as well. We start with a 30 to 40 minute live stream that includes talks from two great speakers, and then we have a little discussion. And then we spend the remainder of the time joining you in the live chat to hear your thoughts and to take your questions as well. Let's get on with some introductions, shall we? I'm delighted to be joined by Dr. Thomas Doherty and Dr. Darina Petrovsky. Hello, both of you. Got their cameras turned off. Come on, join me. There they are. You come in.
Hi, Adam.
Tom is Associate Director of Clinical Research and former Senior Clinical Scientist for pharmaceutical company Eisai. Tom, completed his PhD at the University of Westminster. I think actually that's when we first met. We first met sitting opposite each other on a plane next to each other. And I don't think you realized, but you were laughing along to What They Do in the Shadows on your iPad. You were watching that. And I just finished that series and I was watching too, going, this is so funny. Oh, I can't wait I can't wait to see if he laughs at this next bit. And that was on the way to an AAIC where Tom joined us for the first time on one of our podcasts, which is still there. Go back, listen. Anyway, Tom completes his PhD at University of Westminster, whilst also working with PIs on study design and recruitment, exploring how to best find the right subjects for trials and make the perfect study design, which is what makes him a great speaker for today. Also, I'm joined by Darina, who's an assistant professor at Duke University School of Nursing. Hi, Darina. I can't remember when we first... I remember we also at an AIC as well, I suspect. Although we also know each other through the iStart PIA to support early career researchers, which is another brilliant network. You should all go join. Check out iStart. Her unique background in music training and nursing science has positioned her well to launch her research career with the goal of improving lives of older adults living with cognitive impairment using non-pharmacological approaches. So this is great today. We're going to have two. We talk about clinical trial design. So in the one side, we've got these non-pharmacological interventions. We've got drug trials as well, which hopefully will give us a really good perspective on the two different ways. But let me set the scene more formally. Clinical trial design is more than just a plan. It's where scientific standards meet clinical challenges. Each decision from choosing endpoints to randomisation can significantly affect patient outcomes and the future of treatments. As we push for innovation, how do we balance speed and safety, especially in trials with serious conditions like dementia? And are we designing trials with the patient's best interests at heart, or are we focused too much on trying to make those groundbreaking, achieving results? And how can we improve trial design to meet today's needs and deliver more rapidly? And I can't help but things that the whole landscape of trial design has potentially been influenced significantly since the pandemic and COVID, when needs must enforce us to particularly reconsider some very well established ways we deliver research and how things have changed since then. So how can we improve trial design in the Salon today? Those are some of the questions we're going to explore, but I didn't set our speakers any expectations. They're both going to present whatever they've chosen based on this topic, and Tom is going to go first. So everything I've just said could be completely irrelevant if our guests present whatever they like.
It's pretty relevant, I think.
Brilliant. Well,
I'm going to hopefully cover most of it from a slightly different perspective anyway.
I'll turn my camera and microphone off and over to you, Tom.
All good on the slides, Adam, just so we're sure.
Yeah, we can see your slides. Thanks.
Lovely stuff. Okay, brilliant. So thanks for having me again today, Adam, and thanks for everyone for joining. I just wanted to kind of give my perspective on, essentially focus on clinical trial design in Alzheimer's disease. Obviously, I've been in industry for a long time now. Well, can call it a long time, maybe 10 years or so, and just kind of give a flavour of what it's like from industry, the key points that we want to think about from our perspective and how we approach patient perspectives as well, and also how the whole landscape's changed in the last year or so in terms of disease modifying therapies kind of forces you to have a slightly different approach to your trial design. So just to start out with, as I have to do, as a disclaimer, as Adam said, I'm obviously an employee of ASI, and any content I'm discussing here reflects my own personal viewpoint and not that of ASI itself. As such, I won't be able to discuss any products or anything to do with our compounds at this point. So to begin with, I just really wanted to sort of center everyone on sort of generally where we are in terms of different trials and the different makeup of moving through clinical development. So obviously when you get your compound to begin with, it's come from discovery. It's gone through a whole process of lead identification, compound identification. And hopefully with that comes a nice biomarker package in combination with that. And you move it towards the start of its journey in terms of phase 1. So that's a single ascending dose study, or SAD, and then moving on to a multiple ascending dose study, a MAD study. And typically here, we're looking at healthy individuals that don't require investigation in the patient population. You're primarily focused on PK, safety, tolerability, and finding good doses and a good dose range and making sure the compound behaves in the way you're expecting it to, based on the preclinical and sort of discovery data. The predominance of my focus is more in phase 2, 3, and 4, and sort of that's where the rest of the talk's going to move towards. And these are more sort of biomarker clinical outcomes driven studies. And with phase 2, you're typically looking in the smallest phase 2 at a biomarker study where you're essentially trying to find a dose. And then in a larger phase 2, you're potentially looking at both biomarkers and clinical endpoints in a much larger study to essentially give you a better idea of that dose and sort of confirm it before you move to the phase 3, which is the large registrational study that costs the most amount of money, into the billions of pounds sometimes. And then hopefully if the phase 3 is successful, you can then move into phase 4, which is looking at more post-marketing registry type studies and sort of label extensions, or if you want to sort of change some little aspects to the drug, can be done there. And hopefully that, depending on the scope of those, depends whether you need to go back to 2 or 3 and move on from there. So just so we're all on the same page, there's lots of different things in terms of to consider at each stage of this development, but in essence, the things that I'm going to talk about apply mainly to 2 to 4 in that regard. So I wanted to begin with the patient perspective. How do they approach a clinical trial? Typically, what we've seen historically is it tends to be those that are more well educated, the more affluent, come to clinical trials because they're more aware. However, that is changing recently with the better recruitment methods that have been employed by sites, both more or less in the US at this point, but the proliferation of that is beginning to happen both in sort of Asian countries and also in Europe. But in terms of what the main focus is and the main thoughts that a patient tends to have, I'm obviously generalizing a little bit here, but in the main, you want to understand the likelihood that you're going to actually get this active compound. So the risks that you are taking, albeit sometimes minimal, is the chance of me having the active component 50-50 as it would be in a phase 3, or is it slightly tailored towards it? And how can we, as clinical trial designers, improve that chance? Can we have a placebo group across multiple trials in some cases? Typically, that's slightly more difficult with industry trials, but it's obviously something that you can consider, whether that's an AI-generated placebo group that you're looking at a pre-planned decline, certainly with an Alzheimer's. Not something that's broadly accepted at this point, but something that has potential. And then when we're looking more from their perspective, do we want to accurately understand the safety profile before we move it into patients? So as I said, phase 1, predominantly you're looking at safety, but also early phase 2 sometimes. how do we design the informed consent so the patients are made aware in the best way possible, in the most understandable language, that what they're going to take part in is of benefit both for themselves. And a lot of the time we hear that essentially the subjects that come forward are doing it not just for themselves, but for greater purpose in terms of moving the field forward and actually contributing to the science. And without them, we would obviously not be able to do these trials. So that is a fundamental part of clinical trials. The other thing that has really come to the fore, most certainly with AD trials, even though we're moving earlier and earlier, we're still looking for study partners that have to participate in the clinical outcomes. And also, when you get more progressed subjects, it becomes more important to have those carers or those other halves around to kind of give you a good lead on where those subjects are. I kind of touched on study recruitment as well. We need to be broader in that, and I think that's a big challenge that we're rising to meet at this point. I know the FDA as well is certainly pushing for that to happen, and they've introduced a number of initiatives within new clinical trials that have to hit certain thresholds, or at least have plans to hit certain thresholds. And as I said, the study sites themselves are becoming very good at the proliferation of outreach, and that drives a more diverse population within your trial. And finally, from a patient point of view, the burden of the relevant outcomes, how do we as trial designers mitigate that as much as we can? How do we get the right outcomes that we need at the minimal burden on the patients? And that's always quite a fine balance, because the earlier on, the more you want to be slightly more broad, but then, so in a phase 2, you might want to look for some broader endpoints to potentially show you something slightly more interesting, whereas phase 3, it becomes a lot more circumspect. So you have to have a lot more of a refined endpoint selection criteria, and more is less when you get further down the line a lot of the time. So those are just some sort of points to consider when thinking about clinical trial design from a patient perspective. What underpins most of the discussions in industry in terms of the development of a compound specifically are the 4 R's. So you want to have the right hypothesis. Are we looking to interact with the right disease process? Are we looking at the right compound at the right stage and in the right population for what we think will happen with that compound? Are we also administering that drug at the right dose, whether that's the frequency, whether that's the actual amount of drug we're giving to the subject? So those are all really important considerations that need to happen early. So we need to have a good plan in place and a good broad understanding of the compound before we move it forward. And then finally, as I sort of discussed on the last slide, to have the right endpoints. So a lot of the endpoints discussions are kind of set. I'd say we've used probably the same endpoints for the last 10 to 15 years. There's a lot of discussion around that, and I don't have the time to go into that today, but people like John Harrison do very good jobs at summing up the arguments around the endpoints used in clinical trials very well at this point. All of this, all of the 4 R's are kind of predicated on a fundamental risk that we all take in terms of moving a compound from forward in any point. There is always risk at every stage. You could probably find a reason not to do something with all compounds at some point. However, we all want to find success. We're all striving to improve the disease outlook for whatever trial we're looking at. But one of the biggest ways to do this is to look towards the end. So you start at the beginning of your plan, of your development cycle, by looking at where you want to be. So at the end of the day, you want an approved compound. So you obviously only get there with positive data, with showing statistically significant effects compared to placebo. So the conversations begin at each stage. And for those that aren't fairly familiar with regulatory interactions, it tends to occur essentially before you move to each next stage of development, because then there's a whole host of reviews, cycles around each trial and each outcome of those trials. One way in industry you can sort of have positive, successful discussions. We want to obviously incorporate validated assessments. Now, that's not always as validated as so psychometrically or what have you, but something that the regulators feel accurately measures the disease itself, and other comorbidities, spelled that wrong, so apologies, and how that can limit risk, and known commodities limit risks. So what I mean by that is essentially when we have something that has been used for 15, 20 years, whilst you have all of the drawbacks of those scales and the insensitivity from when they were initially designed, because they've been used so broadly and used so widely, they're something that we understand fundamentally. So moving away from something like that, or something like the CDR, something like the ADAS cog, typically the MMSE is not used in this way, but moving away from that increases your risk. And obviously when you're trying to mitigate any risks, using tools that are not as well validated in something like a phase 3 is something that is essentially inherently risk taking. So is it something you want to consider at that point? Another way to look at this is obviously to broaden your risk sharing with partners, whether that's entering into deal sharing with other companies, or essentially co-developing things with academic partners and shouldering some of the costs, and therefore helping to broaden the perspective of the number of subjects. Also, who you get in to do your trial then becomes a whole different perspective. I can again give another 10-minute talk on academia and industry collaborations, I think. And ultimately, this tolerance level comes down to what your company's like. So whether you're very risk adverse, or whether you're out there looking for new compounds, whether you're out there trying to get push forward as much as possible, sort of alluding to Adam's opening, how much do you want to pursue short-term gains in terms of speed versus unsure outcomes later down the line? So this is kind of my last slide, and I kind of wanted to focus just on AD clinical design itself. And as I said at the start, one of the biggest changes we've seen in the last few years is obviously the advent of disease-modifying therapeutics. So there's a number of those on the market now, and I've just kind of walked through these sort of considerations that certainly we consider at the moment, and certainly something that will be commonplace moving forward as more and more people are treated with these antibodies. So what we have to think about is whether we prohibit them. So do you not allow subjects to take these? If all of these considerations would depend on what your compound actually is aiming to do. So if you've got another anti-amyloid therapy, you would obviously prohibit anyone that has taken an anti-amyloid therapy or is taking. However, that would then reduce your recruitment pool. So you've then got something to balance there in terms of how you approach your recruitment. Do you limit which amyloid therapies are being used previously? We know there's a different range of expected outcomes in terms of PET change, amyloid PET change across that 18-month period, as we've seen from this trial. So do you want to limit some and not others? And do you want to give that choice to your physicians? who are going to run the trials for you. How is that in terms of a normal clinical care? Do you want to, in essence, step in and make that decision for you? Typically, that's not something we'd want to do. And the problem with the amyloid therapies is obviously there's a different profile between them all. So some have titration, some do not. Some have higher safety risks. Obviously, I'm speaking about ARIA here. So that's amyloid-related imaging abnormalities, for those that aren't aware. So some of those safety risks come very early, or some are more spread throughout the course of the 18-month treatment periods. As I said, sort of the rate of amyloid clearance on amyloid PET is kind of important in terms of how if you have that as a baseline level of clearance, how are you expected to see a drug treatment response post that? If you have two different antibodies that are clearing amyloid at different rates, how do you attribute your own drug on top of that to any outcomes that are related to that? Again, going back to one of the 4 R's, in terms of the population, you want preclinical. Is this a preventative medicine? Are we trying to stop any clinical symptoms before it's happening? Or is it something that we're going to prescribe to symptomatic patients to essentially mitigate the progression of disease? And then how are we defining that? So this is something, it's more complex than necessarily you're thinking. So if we obviously have subjects who have a level of amyloid, are we prescribing how much amyloid we're having in the brain? Or if we are looking at amyloid and tau, how are we defining both of those combinations of pathology within the trial itself? And if you're looking at both of those, you're then having a higher screen fail rate because you are then refining and refining and refining your population. So it becomes problematic, it elongates your screening period, but then you obviously need, if you have a tau targeting antibody, you obviously need some level of tau pathology in order to have any sort of target engagement or treatment effect. So lots of points to consider in terms of the study design itself. Are we looking at a 2x2, a factorial design? Some great examples of that are the AHEAD study with lecanemab, or the DIAN-TU study, which is looking at dominantly inherited Alzheimer's. There's also more platform trials coming in the future I'm aware of. And again, are you starting both compounds at the same time? Do you start an amyloid antibody first and then introduce your treatment after some of the titration or safety risk is mitigated or the time has elapsed? Are you then swapping them around? How are you looking at the sequencing of administration? So if you're looking at an antibody every 2 weeks, are you going to give the same another antibody at the same time, so that patient's sitting there for essentially up to 3-4 hours having infusion after infusion, or are you trying to stagger that somewhat? Obviously, in terms of modalities, this is dependent upon your actual mechanism of action or your molecule itself. So is it an antibody, an ASO, small molecule? How are you prescribing it? Is it a subcutaneous formulation or is it an infusion? All of these things are kind of really integral to how you design the trial itself. And then this becomes even more pertinent when you're looking at dose ranging studies. So in the earlier phases of 2A-ish, 2B, you're essentially trying to find the optimum, the sweet spot. And all of these things require even more thought when you're looking at different dose ranges at that point. And then finally, again, I sort of touched on this already, but the clinical outcomes or the biomarker outcomes, you have to really understand what is doing what if you're having combination trials. So whereas before you're essentially just looking for a single outcome and a positive single outcome, now we have to have we have a demonstrated profile for each of the compounds that have come before, and therefore we have to demonstrate an ability of that cohort that you have selected based on all of these criteria to show an increased effect, either synergistically or additively, on top of what has already been shown. So you always have that new bar to beat on the basis of what you've seen before with these compounds. And as I sort of touched on at the start, whether that's a biomarker endpoint, whether it's PET, a CSF measure, even maybe plasma in some point in the future, or if you're looking more into the registrational studies, we'll always have to look for clinical and functional endpoints to some degree, whether that be a preclinical or a symptomatic population. I think it's fundamental to look at in that regard. And all of these things are new, they're evolving, and it's going to be really interesting to see how this moves forwards in the next few years when all of the antibodies come to the fore and become more commonplace in clinical practice. But some food for thought there, at least. And I think that was my last one. So thanks, Adam. If I can hand back to you.
Brilliant. Thank you very much, Tom. That was fascinating. You went into loads of detail and covered lots of points I hadn't even particularly considered about, particularly on drug trials. I missed, I don't know if I missed this. We had a really interesting session a couple of weeks ago on reproducibility. And it was talking, and Rebecca Williams, one of the guests, was making a case for pre-registration of studies, and there was a lot of conversation in that about whether that concerns from some of the guests, audience rather, about it stifling flexibility and that, you know, being able to dynamically change as the trial develops. When you start off on a trial, you define what success looks like, I assume. Yeah. How much accommodation in that is there in a drug trial for what you hear back from participants as opposed to just what you see in, you know, testing?
Yeah. Well, in terms of their feedback on the trial itself, and stuff like that.
I mean, we've seen this, haven't we, with some of the drugs you talk about, that some of the patients report back that this has been incredible, yet when you might do the scans or the biomarker tests or whatever, it's not necessarily showing the same, you know, the level of clearance, say, if in amyloid that you would have liked, yet patients say, I can tell a difference more significantly. That connection between experience and the physical.
Yeah, so typically we wouldn't look at the data. So there's no way of us understanding a biomarker response until the end of the study because it will be blinded. So the only time I think that's sort of applicable is maybe adaptive randomization. So certainly I'm thinking of the lecanemab phase 2. I'm sure there's many other examples where essentially you've got a blinded algorithm working in the background that attributes, looks for the treatment effect at every interval, and then will assign more subjects to the most likely arm to be successful. So therefore you reduce your recruitment, you speed up your recruitment. Sorry, you reduce the numbers you need to recruit, and you speed up the time it takes to recruit those because you're not randomizing to the least likely arms.
So is that the M&D SMART trial at Edinburgh, the Siddharth and Chandran one, I think works like that, doesn't it? I mean, that's on repurposing, but again, in that same way that it's rapid evaluation. Okay, so I had a couple of questions, and then we'll come to Darina as well. Given your experience with clinical and pharma development trials, what do you think are some of the most significant challenges we face now in designing trials that are going to rapidly
evolve? I think a lot of the fundamentals, sort of my last slide spoke to it quite broadly, but I think fundamentally it's going to be showing a treatment effect on top of what we've seen. So if you're trying to have a similar effect using another antibody, are you trying to, and the sort of debate around clinical meaningfulness of the effects that we saw, both not just on a single trial, but across the class of compounds. How do you define a treatment on top of those in terms of its effect, both clinically Biologically slightly easier, I think. So if you have a PET scan and you clear it quicker, or you're also hitting downstream markers. So I'm thinking if you've got a tau antibody and an amyloid antibody, we know some of the amyloid antibodies also hit downstream markers of tau. What we want to see a greater effect on the tau markers because it's a tau targeting antibody. But if you've got other compounds that aren't necessarily as clean as that, how do you show a symptomatic effect or a clinical effect on top of what you're trying to achieve? And to be honest, a lot of the time the regulators are pretty open about having discussions with you and coming to not necessarily an understanding, but they set out their positions quite clearly. And then it becomes a case of, right, okay, so how do we work within that framework to achieve what we need to achieve with our compounds? And so it becomes quite a collegial, not collegial is probably the wrong word, but quite an in-depth discussion around
and just thinking, because obviously one of the things that all the experts are saying right now about, particularly about the anti-amyloid therapies, is that it's unlikely they'll be used in isolation, that they're going to be rather like HIV treatments, that these are going to be, personalized drug combinations that will end up having the big effect. Does trial design get a lot more complicated when you suddenly start looking at, I can certainly see when you start trying to combine things, surely that
so it kind of goes back to when do you start them? So do you administer at the same time? And then the delay that you may have, how do you then account for that with your endpoints?
And different dosages and people,
and it becomes incredibly complex. Luckily,
there's and the population of people
statisticians.
And I can't remember. There's some statistic. Maybe Darina might know this one. The average kind of person living with dementia in their 70s that's got probably 2 or more comorbidities, they're probably on also another 4 medications as well.
And luckily, I mean, certainly the trials that I've worked on, we've allowed those. So we've been pretty broad in our comorbidities, co-medications, concomitant medications, I should say. I mean, fundamentally, when it comes to trials of the future, you want to look at the symptomatics. And typically, I think most of the trials in phase 3 that have just read out have all excluded biologics to begin with, to essentially account for any sort of interaction effects, because you won't understand those within a clinical trial in such a restricted population. Whereas when it becomes more broadly used, how do you account for those? It becomes a lot more difficult.
I mean,
problems we're all thinking about.
I mean, recruiting to a single trial alone with a fairly broad population is hard enough. I can't remember. I've got some stats written down here. I can't remember what the between screen failures and dropout rates and everything else. It's hard, let alone then to also look at people who are on specific other medications or combine them.
Yeah, I think typically we look at anything below 80% screen failure rate as a success. But predominantly that was due to having clinical then amyloid pathology confirmed. So there's always a stepwise approach in the screening process. But I think we got as low as 70 at some point, but that was an exception, certainly.
Well, and that's why I guess what people read in the news and what people hear about this is important. So you've kind of got both sets of narratives going on right now, haven't you? Whereas you've got, wow, this is an amazing breakthrough, this could make a difference, and oh, these are incredibly dangerous, depending on which regulator you want to listen to or which expert you want to listen to. And I think as a participant, the decision then about whether you, I mean, we're slightly going off topic here from trial design, but I think that's an incredibly difficult decision to take then as a patient. Thank you very much.
So I was just going to say, you have to hope your doctor's well informed
about
the latest
exactly. And if, I know most of the people watching this are going to be researchers, but if you're patients, it's kind of, don't be afraid to ask 101 questions. And if you're not, the problem is you don't know what questions to ask. So come back and ask somebody else. What question should I be asking? Darina, sorry, you've been incredibly patient. Thank you so much. And we're going to go in a completely different direction now because we're looking at non-pharmacological interventions and your research on music. But I'd love to hear your talk now. Thank you.
Sure. Sure. Happy to be here. As Anna mentioned, my name is Darina Petrovsky. I am an assistant professor at Duke University School of Nursing. I'm also a professional musician who turned a nurse into healthcare, and I conduct and design clinical trials specific to music-based interventions. So I just had a few slides. I wanted to acknowledge a really influential program that I attended. This is a program in the US. This is called Impact AD. So it's a week-long institute that focuses specifically on today's topic, on methods and protocols for the advancement of clinical trials in AD and ADRD. It's both, it has a fellowship and a research track, and so some of the slides I sort of borrowed from that course and found it to be very, very helpful. I also want to acknowledge my current funders as well. I think I'll start broadly and similar to Tom's talk. There are three primary goals that we focus on for non-drug, non-pharmacological clinical trials. Does the treatment or therapy have a beneficial effect over a comparator group? Is the benefit clinically meaningful? And I'll talk a little bit about the differences between non-drug and drug trials. The second point can be kind of tricky to figure out in non-pharmacological trials. And then lastly, is the therapy safe to be administered? Contrary, I think, to popular belief, non-drug interventions do carry, some of them do carry quite a bit of risk, perhaps more psychological than physical risk, but we do have to think about, is our treatment safe? Does it have does it cause or is associated with any adverse effects? So unlike a model a pathway that Tom talked about, the non-pharmacological intervention or therapy development is a little more complex, I will point out. And there was an influential paper published now, more than a decade ago by scientists from the National Institutes of Health, which is what NIH stands for, which is a biomedical funder of biomedical research in the US. And you see here the stages of intervention development, starting from stage 0 all the way back around to stage 5. But you'll notice that it's in a concentric circle, and there's sort of arrows going from pretty much all the boxes back. And so with this sort of the main message of the NIH stage model is that the development does not occur in a linear pattern, that sometimes when we develop a therapy and test it out, let's say in stage 2 in efficacy trials, we may not find efficacy or benefits. And that sometimes leads us back to stage 0 for basic research to then reconsider and think about, should we improve our therapy? Should we change it? Should we do an add-on? And this can also happen at later stages as well. The benefit of this model is that sometimes we can skip a stage if there's enough, for example, efficacy research showcasing efficacy over a therapy, and then we are ready to test it under real life scenarios in the community, that we can go from stage 1 to stage 3, for example. The other paper that I really is one of my favorites that I have saved on my computer actually compares, does a comparison between drug and devices development pipeline, as well as perhaps non-drug trials. And this is the NIH Science of Behavior Change model, also published by scientists from the NIH, and essentially SOBC, that's what the acronym stands for. And essentially what this, the main purpose of that paper is to say, listen, we have developed many non-drug interventions, but what often happens, as indicated by that second pipeline, is that we think about the intervention, we develop it, we test it in clinical trials stage 1 through 3, and we may or may not find a beneficial effect. If we do find a beneficial effect, we say, great, that's awesome. This thing that we developed worked, but what we don't know exactly how it's working. So we cannot, so we have questions about, okay, if we are to adapt it to the real life setting, what do we want to change about it? Which components do we want to keep? So this paper that came out in 2018 said, okay, well, instead of thinking about, okay, let's just develop the thing that we want to test, so why don't we think about it maybe more so how our colleagues think about it in drugs and devices, thinking about how do we isolate what would cause the change in our desired behavior, think about that firsthand, and go through the stages and have the stages 1 and 2 of clinical trials. Think about how do we, making sure we engage that specific mechanistic target. And then in later stages 3 and 4, we can take that knowledge and develop more efficacious interventions. And then we know which parts of the interventions are actually driving the clinical and meaningful benefits for our participants. Maybe perhaps similarly to what Tom talked about, in design of non-drug interventions, we also think a lot about and have to make several important choices, careful choices in terms of, 1, what is the population that we are targeting? Is it representative of our target population? Is it appropriate to the type of the intervention, to what we're asking individuals to do? And we can think of it in terms of the stages of dementia, as well as whether we're targeting the person living with the disease or their care partner, or both. We also think a lot about duration, frequency, and dose, similarly to drug trials. It's a little bit trickier to capture that sometimes, especially if you are doing work in the community and not in a lab, or inviting participants to come to your campus or to your research facility and partake in the therapy. A lot of careful choice needs to be made in terms of the primary and secondary outcome measures. There is quite a variety of obviously measures that we have used in non-drug trials, and we really don't have a consensus on what really should be using. And there's really a movement right now of person-centered outcome measures, of measuring things that are actually meaningful to the individuals. But to the flip side of that is we really sometimes don't have really good measures that are meaningful, that have been tested in larger trials. So we really don't know if there's going to be movement on those outcomes as we sometimes start the trial. A lot of that goes into sample size considerations. That could be a whole topic of its own, to think about, okay, if we are designing a clinical trial, what effect sizes are reported in related literature. And sometimes we cannot or do not have feasibility or pilot trials that could inform our efficacy or effectiveness clinical trials. I would say control group or selection of the control group is the one of the probably factors that distinguishes drug and drug trials the most. We think a lot about what constitutes a comparator group and what is sort of what is our placebo to our therapies. And then in terms of the control of bias, we run into issues related to blinding of the participants, because obviously if they're participating in therapy or intervention, they would know that they're part of that group, and it's hard to blind those participants. It's a little easier to blind the raters who measure the outcomes of these clinical trials. You may think, okay, well, there's a lot of, right now, especially in the last few years, a lot of focus on drug trials, so we may not be funding or supporting a lot of clinical trials that focus on non-drug interventions. So this is data that I pulled from the National Institute on Aging, which is the Aging Institute of the National Institutes of Health in the US. And they update this figure quite frequently. I think this was updated in March of 2024. So you can see out of a total of 507 clinical trials that are currently funded by the NIA in this space, about 72 trials are pharmacological. So these include phase 1 and 2, as well as phases 3 through 4. But you can see the majority of the trials in comparison are actually non-pharmacological that is funded by the NIA. Most of them looks like the biggest category out of that is the exercise trials, but they also have other trials as well. I believe the one that's the bottom one, that's music, it may be my trial. I'm not sure, but hopeful thinking. In addition, there's also more than 200 trials focused on dementia care and caregiving. So we think that as much as we should focus on treatment, development, and finding therapies for the cure, we also need to find therapies and interventions that focus on those living with the disease as well as their caregiving. So there's a plethora of trials focusing on different aspects of dementia care and caregiving, as you can see here. And then we, at the last category, see sort of lesser numbers of different trials that focus on treatment of neuropsychiatric symptoms, the diagnostic tools and assessments and imaging, as well as the trials that understand the disease processes. As I briefly mentioned earlier, there's a number of non-drug trials. They focus on exercise, cognitive training, modifications to the diet, sleep, combination of different non-drug therapies, and many different ones. The one I want to point out in the others category is the light therapy, specific blue light therapy, and that's the type of research that I started doing when I first entered dementia space, as well as work in transcranial magnetic stimulation. I want to touch briefly upon the distinct challenges in relation to non-drug interventions. One I mentioned is the effect size. When designing a brand new intervention, can be especially challenging to think about and base our literature review and base our effect size calculations for our sample sizes on existing trials. Thinking about, fidelity of the intervention, and that has to do, and it influences the rigor of the study. Thinking about, okay, if we are administering the intervention, do we administer it the same way every time that the participant receives it? Thinking about design issues, I mentioned this sort of thinking about what is the perfect control group. One of the books that I highly recommend for those of us who are doing behavioral and social intervention research is by Laura Gitlin and Sarah Saja. They have a really wonderful chapter in that book talking about considerations and the different types of control groups that we could potentially design that are really driven by your research question. We also think a lot about the infrastructure. How do we ensure that the outcomes are measured in the same way and that they're measured in a blind way? Intervention delivery as well as training sites, so we can have interventionists or individuals who administer the intervention. How do we ensure that they're trained the same way? How do we ensure that they deliver the intervention the same way with each participant? And thinking about maybe similar to drug trials as well, these trials are very expensive to conduct. We need to have both the infrastructure as well as the staff knowledgeable about, one, how to work with individuals with dementia, and be familiar with how to administer our therapies in the most rigorous way. I'll briefly mention my own work. So I started with conducting feasibility music listening intervention focused on reducing sleep disturbances in older adults living with dementia. This was a protocol I published right in the middle of COVID, where I talked about the design features, the control conditions, and all these things that are thought about when designing this particular trial. And you can see that about 3 years after that, I published the results of this feasibility RCT. We unfortunately were not able to recruit the full sample. You know, keep in mind that this was when I was a trainee. I was a postdoc at that time, and it was right in the middle of COVID. However, in terms of our sample was quite diverse. And so remember, this was a sample based in the US. And we found some movement in the intervention group as compared to the control condition. And I used the results from this feasibility trial to write a larger grant, which I received from the NIH, to then take the feedback from the feasibility RCT, and I designed a larger RCT to look at the efficacy of a music-based app for sleep in this population. And that work is currently ongoing. And as I get older, with each passing year, I learn more and more about how to design rigorous clinical trials in non-pharmacological space. And so the study that I designed back in 2020 taught me a lot. And so now my thinking, and as our science and as our thinking evolves over the design of clinical trials in non-pharmacological space, you know, I believe the rigor of my work goes up with each year. Here's a link for me if you'd like to get in touch with me or learn more about my work. I'm really excited, and thank you for the opportunity to be here.
Thanks, Darina. God, we're running terribly on time. I'm a rubbish host when it comes to timekeeping. But if you're watching and you've got questions, please do post them in the chat, because I don't think we're going to have a great deal amount of time to join you in there. But let me just rattle through a couple of questions now to round up, and we will still try to get in there. But if you post questions in the chat, I will put them to Darina and Tom to make sure you get an answer to that. So, Darina, your background, I mean, that was a brilliant talk, and it complemented Tom's really nicely, because you went into lots that obviously Tom didn't. We've got, I can see your screens in your glasses. It looks like you've just joined the Matrix. It's all gone across. It's awesome. No, it's really good. Your background is quite unique in combining that kind of music training. I didn't realize you were a musician. I mean, it makes sense you'd be a musician first and then became a nurse. How has that kind of interdisciplinary approach influenced your perspective on trial design? Do you think it has?
Yes, I think so. I think there is more and more work coming out in this space that really requires me and others to partner with other disciplines, such as music therapy, music perception sort of world. And they definitely, those communities may not have thought about how to design sort of clinical trials and all the things that go into the design. So absolutely, I think right now it's sort of a requirement for me to involve different members of my team to have different perspectives, and that has really enhanced my work.
Are we reaching a point? So the outcome measures for drug trials, for example, have been quite well established for quite some time. There's hundreds of them in different spaces. Are we reaching a point in arts-based research and therapies where we're kind of starting to have a standardized approach for defining the success of an intervention, do you think? Or is each trial still doing its own thing?
Yeah, I would say we're actually moving in the opposite direction. So I mentioned the whole idea of meaningful, personally meaningful outcomes. I think that has definitely sort of have come about in the arts community, given that a lot of sort of mechanisms of action of arts-based interventions do not very nicely play with, cognitive outcomes. We think that they have effect perhaps more on sort of neuropsychiatric behaviors, as well as, moods and emotions. And those types of outcomes we have not seen, and neuropsychiatric symptoms, of course, we have seen looked at with drug trials, but it's kind of, I feel like we're all over the place. So I think the community, we're trying to think about how to standardize some of person-centered meaningful outcomes. There are some that are broad enough to be used and would have an effect in other types of arts-based interventions. But having learned, when I attended that institute I mentioned, I was blown away by the consistency of outcomes in drug trials. And that has definitely not been the case in my work or in others' work as well.
Yeah, I think some of our previous contributors of blogs and things are working on exactly that. In fact, I know a researcher, Victoria Gabb's going to start writing from us, and she's from Bristol, looking at she originally worked on sleep research, but they're doing exactly this right now to try and come up with an international consensus on what defines success for some of these interventions, which is never going to be easy, I think. So just jumping on to a couple of my generic questions. Let's have a look through the list on what we've got time. Technology. How is technology changing trial design? It's changing massively, rapidly, particularly in drug trials with the use of machine learning and AI, I guess, to interpret data. Is that the case, Tom?
I wouldn't say necessarily to interpret data, but it would be definitely to select patients quicker.
Right. Okay.
It's interesting because essentially a lot of what we're trying to do is predict the more expensive assessments. Certainly, so I just published a paper on predicting centiloid values via blood-based biomarkers across the spectrum of AD. That's one avenue that we're applying to clinical trials in the future. I think in terms of technologies as a whole, you can go down the whole collection, eCOA. Certainly we did more remote testing through COVID, and that's something that I think is going to be here to stay. And other sort of more operational aspects like SDV, remote and stuff like that. Yeah.
What about in your space, Darina?
Yeah, I would say so. I think more so, I've seen a lot more machine learning used to identify patients, so similar to what Tom's saying, using EHR and hospital records and community records. Definitely in the delivery intervention as well. I see a lot more telehealth or telemedicine approaches being used, especially to reach those in rural communities that may not have access or may not want to travel or too expensive to travel to our academic health centers. I would say that's
so that's kind of in the delivery. Yeah, I can see that. I don't know. Are we moving? This is just showing my ignorance, so apologies for this. So I don't know everything about everything. Are we moving to a point where, and tell me if this is already happening, where you can already go so far in modeling a trial based using kind of machine learning and previous trial results and the data we already have, that you can model this out before we ever taking it anywhere near patients or people?
You can. So CPADs, the CPATH Institute, essentially have been doing that for a while. And I think the limiting factor with it is the acceptability for a registrational study. I don't think it will ever be acceptable at this point in time. It may be in the future, but certainly regulators are not quite okay with having an AI-generated placebo.
Well, it's whether you kind of, and whether you can jump some of those stages, I guess, with this. Some of the stats I have here that, where are we? I've got a stat about some of the COVID trials that the COVID pandemic accelerated adoption of decentralized clinical trials with an increase of over 50% between 2021, and they reduced the time by, I've got a whole page full of statistics, so apologies.
Well, I think with DCT as well, as Darina was saying, it kind of helps broaden your patient population as well, and certainly serve those underrepresented populations that don't make the way into the trials.
Those in-person visits.
Yeah.
Yeah, and an estimated 70% of drug trials in 2021 included some form of RWE, some real-world evidence to support their application, which is good. Which I think we want, we should be moving towards that point. We're out of time, I'm afraid, and we didn't even get to chat, but that's good because our live audience has dropped off a little bit. But thank you very much to those who've stuck with us for the full hour. I gave you a chance to ask questions. You didn't take it, so I don't feel too bad. But honestly, just because the live stream has ended today doesn't mean that the conversation can't continue. If you're watching this on catch up, please do add your comments below. Tom and Darina are both members of the community, and if you tag them, if you do the little @ and start typing their name, it'll come up. You can tag them into any questions you post in the comments against this recording, and they'll get a notification, and hopefully we'll jump back in to answer your questions if you're watching this on catch up, or if you've got experience of trial design and want to make some points of your own. If you agree with Darina and Tom, disagree, have a different perspective, have some new ideas on how we can improve the trial design in future, please do post those in the comments. Thank you very much, Tom and Darina. It's been brilliant. I'm really pleased. I can't remember if some of you were back next year for some of our next year's talks, because we're planning right through to next September, but I hope you are. And thank you very much for joining us.
Thanks for having us.
No, that's all great. And I hope to see you at Alzheimer Europe or ADPD or one of the conferences that I'm already starting to plan for.
You see me again soon, I'm sure.
Have a great rest of the day. And thank you very much, everybody, for watching.